Hello everyone, my name is Qingxuan, and today I'm going to show you how to simulate the HSQC spectrum using different resources and also how to compare this spectrum. So at first, you need to have two files. The first one is a protein sequence file, and the second one is a PDB file. So we'll start by simulating the spectrum using the protein sequence, by searching from the BMRB database. So just type two-letter code BB, and then we can upload our sequence file and make sure we change to the one-letter code sequence. And then choose the sequence file, and then you can click the plus button. And then you can see for this part, the scores. So shows how similar your sequence is to the entries in the BMRB. So if it has a higher score, it indicates a better match. So you can choose the top one for the simulation. And then you can click the Simulate 2D Spectrum button. And then choose the N-HSQC. And then here's a simulated spectrum with the labels. We can simulate another spectrum using the PDB file. So you can just type capitalize C and CS. And then you can, if you have the PDB file, you can upload directly. And if you don't, you can use a quick ESM fold, and you can enter the sequence, and it will help you find the PDB file. You can type your email here, your name and your email. And then click send to server. And so you will receive an email when the job is complete. So if the job is complete, you can download results. And then you can open the result. And then you click OK, and it will pop up the resonance list. And click the simulated button and choose N-HSQC. So you will get the second spectrum. So before we do the peak matching, we will go back to the first spectrum and use a two-letter code FA to save this file. Save, and then we can click the Control A to select all the signals, and then type A, capitalize D to remove all the labels. So if we zoom in a little bit, as you can see, the labels are removed. So this makes the comparison more cleaner because we want to assign labels based on this spectrum. And then we can do the peak matching. So just type two-letter code, capitalize RP. And the target spectrum will be the left one, just which we already removed the label. So you will see some peaks are already assigned and others are marked with some question mark, but with the candidate assignments. So for this unsigned peaks, we can use a Pine assigner to help us to assign them. So we can select one peak. For example, I select this one. And then I use the Pine assigner. The two-letter code is PR. And update. So it will show the top candidates for this unassigned peak. So the Whiting 70 is the top candidates and with a score 1.15. And then, but to verify, we need to use a peak list. So just type LT. So just go to, so we just to check if it has the Whiting 17 already assigned to other peaks. So if it's not, that means we can use the Whiting 70 for this peaks. So since there's no Whiting 70, we can assign this peak by this one. So you can just click assign. So you can repeat this step for each unassigned peak. The only problem is the live spectrum is simulated using the sequence. So it will include some extra peaks from the side chain, not just the backbone. And some assignments will remain uncertain. So just like for this region, as you can see, it has some side chain peaks, but the reference spectrum doesn't have this assignment. So in this part, the assignments will remain uncertain.